Science Cafe. Hope that this will be the first of many great talks that we have, and so it's my pleasure to introduce Dr. Jed McCoskell, who is a physics professor at Wake Forest. Dr. McCoskell got his bachelor's degree from MIT. And
Thanks a lot, guys, for having me here today. But I wanted to talk today about how biology, physics, and visualization can be tied together. Many of you may be involved in the scientific visualization program. Others of you may be involved with biotechnology. And physics plus biology, in a lot of ways, makes the backbone of biotechnology. Mr. Howerton is my slide advancer. So how do physics, biology, and visualization relate? Do they? I think so. Here's how they relate in my mind. Physics deals with forces and things like that, as you know, if you've taken physics already. There are a lot of things you can do on the macroscopic scale, like balls rolling down hills, or boxes being held up by strings and pulleys. But also at the molecular world, there are forces and matter and equations that can govern things that happen at the molecular scale. Biology deals with living things. You have dissections and frogs and all kinds of stuff like that, organs, ecosystems, again, macroscopic things. But biology also can be shrunk down to the molecular scale. And it's when you shrink physics to the molecular scale and biology to the molecular scale that you really can see that they come together. Okay, now, scientific visualization allows our eyes to aid our understanding. Your eyes are powerful tools. They can read text and they can gain understanding that way. But when you visualize something properly, you will be able to aid your understanding even more. And so the one video that I'm going to show you today was one of my first examples of how visualization can aid my understanding through my eyes. My research at Wake Forest involves all three, so I think that the biology and physics at the molecular scale really come together, and I think that visualization is the key to understanding those things, especially since they happen at a scale that not even microscopes can see. So you never can get a visualization with a real instrument. You're always going to have to rely on computer graphics to understand really what's going on. Can I have the next slide, please? Okay, and you can just click through until it shows everything. That way, I'll have everything pulled up. Whoops, go back one. Sorry, this will be the video from Paris, France. Does it go back? Can you hit the back button? And I'll just talk a little bit before we show that video. Go back one more slide and we can see Mr. Sagan. There we go. Oops, and forward one. There we go. Okay, there's Mr. Sagan. Mr. Carl Sagan. How many of you have heard about the new cosmos that came along? Who's the host for the new cosmos?
Neil deGrasse Tyson.
What? Yes.
Neil deGrasse Tyson.
Neil deGrasse Tyson. And he did a fabulous job. This was the host of Cosmos when I was your age. Okay? In fact, it aired in 1980, so I was only 8 years old when I first saw these. And they really were impressive. I liked seeing the billions and billions of stars and thinking about galaxies far, far away, thinking about different creatures that might live on different planets or the moons around some of the planets in our solar system. It was very exciting. But my favorite episode was episode 2. And the reason it was my favorite is because inside that episode there was a computer-animated DNA molecule. Okay, the guy who animated it had worked at Microsoft a little bit, and he was a young guy with some computer science PhD, and with Carl Sagan and the other people who produced Cosmos, he made a model in 1979 and 80 of DNA using computers. And he even modeled 2 proteins. So if you're in biotechnology, you know that proteins are the same kind of thing as enzymes. Proteins and enzymes do all the work inside of a cell. They get everything done, they can operate like machines, they do stuff, And so these were, this blue one here and this purple one there were doing something to what you notice in green and yellow is the DNA molecule. They were doing something to it. And I was fascinated as an 8-year-old because one of them was called polymerase. That's this guy right here that has this open mouth. And they were showing the video how it was kind of going like this and chewing around, and every time its mouth kind of closed, it stuffed a new little piece of DNA in, and it was making a new strand of DNA. I thought that was fascinating. I also really like this purple one. The purple one was tearing apart the DNA into the 2 strands. That's called helicase. And believe me, I thought this was good stuff. When I was in my little HAG program in 1982, I did my first report on DNA. And I made a little model using some elastic and some cardboard, put together a DNA molecule. And I remember the thing that I thought was the most interesting back then, I was 10 years old, about DNA was that if you stretched out the DNA in one cell, it would be 2 meters long, taller than me now, and way taller than I was back then. So I thought that was fascinating that you could take DNA and pack it up into the tiny nucleus of a single cell, and yet you could stretch it out. I was like, wow, that's like magic. So I was kind of hooked on DNA. I was kind of hooked on scientific visualization of things that happen inside of the cell. And I just was waiting for an opportunity to really find out more about this. Well, my opportunity came when I was a graduate student at Berkeley. And as a high school student, I got to go to a summer science program. How many of you guys think you'll ever go to a summer science program, or you've already gone to a summer science program? Raise your hand. You're going to spend your summer doing science. Oh, come on. It's not that bad, actually. You get to meet people who are a lot like yourself, and it's a lot of fun. I had a great time. I ended up going, you can put your hands down, I ended up going to Washington, D.C. with this summer science program. And I stayed in touch with the group that ran the summer science program. And when I was long after high school, I was already in graduate school. This same group that organized it had a party down in Southern California. And it was fun, because I got to take my friend's car. He drove me, and he was a business kind of guy. And he had this red convertible, and we pulled up to this big, hotel in Southern California, and there were all these impressive-looking people. The governor of California was there, his wife, the president of Pixar Studios was there, and I was just like, wow, I get to go to this just because I was in a summer science program? So I walked in, and I was there on duty to help with this nice little gala, they called it. And I was taking people's coats and stuff. I mean, I was just a 20-year-old kid. And I got to take the coat of the Pixar president, and he started asking me what my PhD research was on. And I said it was on biophysics. And I was studying proteins inside of a cell. He said, oh, well, I have a great video for you. I was just the judge of a scientific visualization group of people from Paris who entered their scientific visualization in a giant contest called SIGGRAPH. How many of you have heard of SIGGRAPH? You've heard of SIGGRAPH. Are you part of the scientific visualization program here? Okay, well, that explains it. It's kind of like the Cannes Film Festival for any kind of computer animation. And if you win something at SIGGRAPH, it's almost like winning an Oscar in that community. And this particular video had won one of the entries because of its beauty and its educational content. And I'm going to show you that video. Now keep in mind, this was SIGGRAPH 1997. Were any of you born in 1997? Okay, so this is like the year of your birth. These videos are old. You're going to see, these ones are from 1980, and if I showed you those, you'd even laugh even more. But the videos that I'm going to show you now, they're definitely old. I mean, they are really old. They're the long gone days of computer animation. But they're still very educational. And this particular one is 15 minutes long, and I have showed it literally hundreds of times all around the country. I have become a salesperson at one point for this video, which I used to retail when I bought it for $126 if you wanted both parts of it. And I paid my own graduate student stipend money for that video because I thought it was so good. Later it was released on DVD, and I think it was only $30 or $25. But I was glad to get it first. So I'm going to show it to you now. It's called The Voyage Inside the Cell. And we'll just see what we can learn through scientific visualization about the physics and biology inside the cell. You can start it up now. So it's this studio in France, and they have something that's similar to our NIH. And I'm sorry, it's very dim. It's so dim that we might not even be able to see it very well. But okay, you can kind of see it. Let me stand at the back of the room and see how much you can see. Basically, there are cells here. Each one of them is kind of a sphere. Okay. And maybe we can kill the sound. Is there any way you can kill the sound? Because she's just chit-chatting, and I'll do the chit-chatting. So each one of these cells is kind of like maybe a white blood cell that you've collected from somebody's blood. You've spun down in a centrifuge the red blood cells since they're heavier. They have maybe some iron in them, makes them heavier. Now you have the white blood cells, and they have these little things on the outside, these little spikes. Those might be ways that the white blood cell can interact with its environment. And on the surface of that cell, you'll notice coming into view are these little heads. Okay, and those are the heads of the lipid bilayer. And a lipid bilayer is what creates this membrane all the way around the cell, and it's a semi-permeable membrane, as you probably learned about in one of your earlier classes. You can see this blue molecule can get through, okay? But other molecules won't be able to. Thanks for turning down the sound there. Because these tails of the lipids, they are like grease. And you know that salad oil doesn't mix well with water. And something that's watery can't really go across that oily layer. But some things can. And the thing you saw go across is a hormone molecule that has some oily kind of character to it. Now this is another hormone molecule that doesn't have any oily character to it, so it can't get across the semi-permeable membrane. That doesn't mean the signal can't get across, because as you see here, it binds perfectly to the top of this little guy, and it sends a signal across the membrane. Now what is that little guy? That guy is a transmembrane protein. Again, proteins do everything inside of the cell. They do all the work. And this particular one releases a signal of small molecules that then bind to this little blue thing. You can barely see it. That's called the endoplasmic reticulum. And when it binds to that, it sends a signal through the endoplasmic reticulum membrane and releases this cloud of calcium. Calcium is just a single atom, okay? And inside of liquid, it becomes an ion charged up with 2 positive charges, but that can be a signal to the rest of the cell. And that signal can spread very quickly because the calcium is so small it can spread. Now there's a third way that things can go across the membrane. We're looking at the underside of the membrane, and what looks like it's a disco ball is actually the assembly of these protein molecules called clathrin. They're made of shown here in blue, they're made of 3 identical arms that when those arms get together, they can form the little holes that you're seeing, kind of like a soccer ball. And when they get together like that, they pinch off a piece of the membrane, kind of like if you were to blow a bubble in bubble gum. You could blow it and then pinch off the little bubble. That's kind of how they do it. And that's how big things get across. Now, this thing looks crazy, but all it is it's the mitochondria, which is, as you know, the powerhouse of the cell, which is why you can see these little electric currents coming across it. When we say powerhouse of the cell, we mean that it takes the food that you would eat and turns it into useful energy for the rest of the cell. What is that useful energy? There's 3 letters. ATP, that's right. Now, if you were in this room several years ago, you would have heard a bunch of Atkins students chanting the following with this little guy right here. They'd say, get your ATP and do your JOB, okay? Which means that this guy, which is a motor protein based on the little motors that you just saw carrying along the mitochondria, it's called kinesin. How many of you have seen a movie where a kinesin molecule carrying a vesicle walks down a microtubule? Raise your hand if you've seen that movie. That's the other movie that I was going to show you, and I'm sorry, since very few of you have seen it, that I didn't show it to you, so I'll rely on Mr. Howerton to go ahead and show that to you, because that's a very famous movie that came out in 2007 or 8, and it's actually very high quality. You'll enjoy it. Not like these cheesy animations you see. Now, this burning sun is the center of all the microtubules. Does anybody know what it's called? No, it's the center of the microtubules. This is the center of the cell, and that is the nucleus. Centromere or centrosome? That would get you the points on a test. The centrosome is where all the microtubules start, and if you remember, the mitochondria was inchworming along one of those microtubules. So those are like the rails. Now here's our little hormone molecule that had slithered across the membrane. It's now going through a hole in the nucleus, and that hole is called the nuclear pore, and there are many of them. And inside of the nucleus, you can see, of course, wrapped around these little yellow spools, the DNA. Okay? Those yellow spools are called the nucleosomes or histones, and these little rods are kind of holding up the nuclear structure. They're part of the cytoskeleton inside the nucleus. Now, what is the purpose of this hormone molecule? Okay, it was coming across the cell, slithering across, went through the nuclear pore. Well, it turns out that this little hormone molecule is a growth hormone, like you might give to cows to make them have more milk or to have more meat, growth hormone. And there's a growth hormone receptor, shown here in pink, that has a perfectly suited binding pocket for that hormone. When it binds the binding pocket, kind of like under its arm, then it's able to make a dimer, or you can think of it as a duet. It joins up with another identical protein molecule, and then when the two of them are together like this, they have enough strength to grab onto the DNA. Okay, so this is all physics. Okay, even though it's happening inside of a biological system, it's physics. So it's physics and biology, and you're seeing it because of scientific visualization. They have now recruited a molecule called RNA polymerase. But before we show you what RNA polymerase does, let's get a little refresher on DNA. I know you guys know what DNA is. It's like the most commonly understood biological molecule. It has two helices, shown here in dark blue and light blue. Between the two helices are these little ladder-like rungs. And if you were to look at all the atoms, they would look like this once you filled them out to their proper size. That's the bumpy nature of DNA. And you can see that if you don't color it per atom, you just color it per strand, it would look kind of like this, and they've simplified it into kind of looking like a double corkscrew. Okay, so that's how they get the picture of DNA. And there it is now again, that's RNA polymerase. What is RNA polymerase doing? Well, as we are in a library here, you can imagine that some of these books, the librarian doesn't want you to check out through those doors, okay, because they're precious. So if you wanted to see what was in the book, you could scan an image on one of the scanners or on a copier and maybe get a temporary printout of it and take that home with you wherever you wanted to study this material. Okay? Now, in the days of electronic articles and stuff, this is not as big of a deal. But you can see that's what's happening here. This DNA is being copied into a temporary copy of messenger RNA, and the messenger RNA is allowed to get out the door, which is, of course, the nuclear pore. Once outside of that nucleus, it can go with a ribosome that makes the copy into something useful. Okay, reads the information, makes something useful. In this case, it has made a DNA polymerase. So if RNA polymerase makes RNA, what do you suppose DNA polymerase does? It makes DNA. And this is the same molecule that Carl Sagan told me about when I was 8 years old. Okay, now we have a little bit of a better picture here in 1997 of what it looks like. In 2014, we have an even better picture of what it looks like. So you could make an even more accurate visualization of this. But essentially, it's always going to do the same thing. It takes one strand of DNA in, splits it apart, and fills it in so that there's two new daughter strands. Each daughter strand, of course, has half of the mother strand. Okay? Once the new DNA has all been made, all of the DNA has been copied, it has to be packaged up around these little histone molecules. Okay? The histone molecules look like little spools, and the DNA fits perfectly around. Then there are these little cocktail weenies, little sausages. We don't really know exactly how they look or how exactly they work, but they pull together loops of these DNA spools. And the loops themselves get wound up into bundles. The bundles get wrapped up into giant sausages, and the giant sausages are what you know as chromosomes. X chromosome, Y chromosome, all that stuff. We've got all these chromosomes, 23 pairs of chromosomes inside of our DNA. And when it's time to divide, there are identical copies on either side. And what happens next is amazing. The centrosome that we saw earlier, that sun-like thing that where all the microtubules start, actually splits into two centrosomes and duplicates itself. Without the need for DNA replication, it could do it all by itself just by adding new building blocks, which of course come from DNA themselves, but it's amazing how it kind of operates as a second way of duplicating. Then the two centrosomes are on opposite sides of the nucleus. The nucleus disappears. All that nuclear membrane that had the nuclear pores in it. Okay, so it leaves the DNA totally exposed, but by then the DNA has already formed chromosomes, so it's safe. The chromosomes get all lined up by these rods, which are the microtubules, and the microtubules get hooked into the side of the chromosomes at a point called the kinetochore. Okay? The kinetochore is full of motor proteins, not unlike this kinesin guy. And so the kinesins are trying to walk down the microtubules, but they're still connected to the kinetochore. So they're pulling this way, they're pulling that way. It ends up looking like a giant tug of war between the two centrosomes, one on the right, one on the left. And once all the chromosomes get lined up right in the middle, then the sticky glue, the molecular glue that was holding the chromosomes in the middle, breaks apart. Okay? And that's happening right here. And now all the DNA on one side goes one way, the other side goes the other way, and you have a perfect copy of the DNA in both sides. So one cell that was once just a cell by itself has now come together and become two, and the membrane on the outside starts to get squeezed in, again by motor proteins, making a cytokinetic ring all the way around, and it squeezes down until these two things just pop apart, and now you have two cells. Okay? And that's happening in your body all the time because you're constantly losing cells. Cells are constantly dying. And if this wasn't happening, you'd be dying. Okay? And when it stops happening, you will die. That's why we get old. That's how we eventually die. Okay? So this is the story of life inside of a cell from the input signal of the growth hormone all the way through to the growth of a new cell, okay, from one mother cell to two daughter cells. And I think that still gets me excited. Here we are, what is this now? 17 years later, okay, from when I first saw that movie. I've showed it hundreds of times all over the country, all over the world. And here I am now showing it again. I'm showing it to you in its entirety, probably for the first time in several years, because it's so outdated. But I thought you guys could enjoy it, and plus, that was the only one I could transfer over to that computer. And